Good day, everybody. The topic of today's lecture is immunobiological preparations, and very often you name them as just vaccines. But please remember that not only vaccines are there, and we have set of different preparations used in medical practice. So let's make it clear, because actually the topic seems like very easy for you, and very often students say, I know that, I will not even refresh my knowledge, I will not read it. But it's one of the main problems in exam. Very often you're confused, you cannot identify, differentiate, or explain what is it, how to use. So if you remember in the very first lecture, very first lecture, I showed you three main reasons, the slide with three main reasons why to study microbiology. And number one was to become a good doctor. So, of course, now it is your task to become a good doctor. You need to study, to prepare, to read, to listen, to have a pack of knowledges. They will allow you to get diploma and become a doctor. But, of course, it's not really enough, because when you will get diploma and you will become a doctor, you need to keep that label of a good doctor valid. You need to be up to date. And of course, you will need to refresh your knowledge regularly, but any doctor, any really good doctor, should be able to accomplish that triad of a good doctor. I call it like that. You need to know how to diagnose disease, how to treat, how to prevent. And of course, for every item, we have a specific preparation. We have diagnostics preparations, we have treatment preparations, we have something to prevent diseases. So in classic way, we have three main sets of preparations. Vaccines. Vaccines are used for prophylaxis, always prevention of the disease. We have immune serum, immunoglobulins, preparations used for number two, for treatment of the diseases, number two, treatment. And we have a set of preparations used for diagnosis for number one. We have two types of diagnosis: in vitro in laboratory, and in vivo or in human for skin tests. Anything labeled with diagnostic cum, it is for lab diagnosis. If you have IN at the end, toluene, brucellin, and some tuberculin, it is for injection, for injection, for allergic skin tests. Later in this lecture, I will explain in more detailed manner. But now, I said already, even for a few times, that we have a lot of classifications of immunity. And acquired immunity can be classified into two more types: active and passive. And if acquired and innate is something easy for you, you can explain easily what's the difference. Then active and passive is something like a Chinese wall for you. It's easy topic. For me, it's very easy, but you cannot remember, explain, apply active and passive immunity. So again, that memory with associations. Try to forget about, if you will be asked what's the difference, forget about medicine and about microbiology. Just try to understand who is active, who is passive. Whom can I call as an active student and whom one is a passive, or active sport player and passive sport player. And when you're thinking like that, you're always given a good and fast answer. Active is someone who always ready, reading, answering, always there. And passive, some student who is sitting and waiting for a miracle. The same is here. We have two types of immunity: active and passive. The main difference is in activity, as it is obvious from the name. Active acquired immunity is the type of immunity when your own cells producing immune response themselves. They can produce antibodies, they can activate memory, they can activate a lot of all chains of immunity. And of course, active immunity happens after a contact with an antigen, with a particular antigen. So active immunity is the immunity produced by your own immune system in the response of the to a contact with the antigen. Your cells will work hardly. They will activate all chains on immunity, of immunity, they will produce antibodies, they will activate cells, everything will be done actively by your immunity. And what is passive acquired immunity? Passive immunity is some immunity which is provided by someone else. It is type of immunity derived by external antibodies. Your immunity will do nothing. Your cells didn't have a contact with antigen. They have no info about that antigen structure. No memory will be formed, nothing. Passive immunity is immunity provided by or driven by external antibodies. That's all. If your immunity given your immune response, active. If someone else provided, passive. So, later again during the lecture, I will try to make it even more clear. But both types of immunity, active and passive, can be subclassified into two more, natural and artificial. And as you can guess, 4 combinations are possible there. We can have active acquired natural immunity, we can have passive acquired natural immunity, both can be artificial. So to make it easier, to make it more clear, again, a lot of things in microbiology and medicine are much easier to remember if you understand. If you will understand what is it, you will remember it much easier. So let's think. We have 4 combinations, as I said. Natural, active, acquired immunity. Just try to think, what is it? We know that acquired is something during the life, after the birth. Okay. We said that active is doing by your own cells, by your immunity. So you should have a contact with antigen previously. Okay. And what is natural? How are we getting our immunity naturally? Of course, here we need to talk about disease. Natural, active, acquired immunity is an immunity obtained after a past disease. It's the very first known, described, and easy way. You have a disease, your immunity is activated, you had a contact with antigen, infection is clear, memory is left, you have an immunity. Number 2, artificial, active, acquired immunity. Again, acquired, everything after the life. If it is active, means your own system will produce antibodies. It will be activated. What is artificial? What is artificial way for pathogen to invade your body? It means it will go not through natural inhalation, ingestion, or someone will give you that antigen artificially. And who can that be? Of course, here we're talking about injections by medical staff, by medical personnel. So artificial active acquired immunity is an immunity induced by injection of antigens. And later we will talk about vaccines and toxoids. So doctors will induce artificial immunity always. Number 3, third possible combination, natural passive acquired immunity. Again, try to think, what can it be? If it is acquired, it's after the birth, seems like. Passive means antibodies will be given, no contact with antigen, and natural. How naturally people can share antibodies? And if you will say donor, donor is not natural way, because we don't have an option to connect with the donors naturally. It is artificial blood connection, but who have common blood circulation? One common natural blood connecting channel. Of course, here we're talking about pregnancy. That's why I said somehow, because during the pregnancy, it's not after the birth, but it is acquired immunity. It's not given by nature. Mother sharing antibodies through placenta. And if you remember again past lectures, IgG are involved here. Immunoglobulin G can pass through placenta and give immunity for a kid. Later after the birth, it is possible to share IgG and IgA through breast milk. And during breastfeeding, mother will donate antibodies to baby. And the main point here, baby didn't have a contact with antigen. No memory is present in baby. And that immunity will be kept only while mother sharing. When that connection is terminated, baby will lose an immunity. So why that immunity is good? Because it's easier for immune system of the baby, no contact, no overloaded, but kid is protected. It will have some time, some reserve of time to become stronger, to get a good condition, and later when natural passive immunity will be lost, of course kid will be able to produce its own antibodies, its own immune response. And the last type of immunity is artificial passive acquired immunity. Again, I said that artificial is always related to the doctor, but if it is passive acquired immunity, artificial, then of course we're talking about infusions of serum, immune serum or immunoglobulins. Later, again, I will describe it in more detailed manner. One more classification of immunity is according to the target. It can be antimicrobial or antitoxic. Antimicrobial means antibacterial, antiviral, antifungal, and antitoxic means against toxin produced by microorganism. Again, it seems like obvious and easy, but why that property is very important to know and to understand. Because if you will have antitoxic immunity, you will not be immune against microorganisms who produce the toxin. And you still will be susceptible to disease caused by microorganisms. The perfect example here is diphtheria. We all are vaccinated with diphtheria toxoid, DTP or some other vaccines, pentavalent vaccines you have. So it provides immunity only against toxin. But people are still susceptible to throat infection. They can act as carriers. They can carry pathogen. They can have some abbreviative disorders in throat, but they will not be harmed by a toxin. The same is against microbe. If you have microorganism as the part of the vaccine, you will be immune only against microbe, but not against toxins produced by them. So what is vaccine? You all know what is vaccine. Now especially you know what is vaccine, you can explain, but we need to have some definition. Again, I prefer some common worldwide definitions. WHO defines a vaccine as a biological preparation that improves immunity to a particular disease. Improves immunity. Vaccine itself is not a drug. It cannot help in curage or some, but it helps to improve immunity. So vaccines usually contain antigen, and it contains agent, microbe, which resembles a disease-causing microorganism. And very often it's made of weakened or killed form of the microbe. Or it can contain only some most immunogenic superficial surface proteins. As the type of vaccine, we can have toxoids, weakened toxin. So the very important thing here, vaccine is contain antigen. It is a preparation, IB immunobiological preparation, contain antigen. It's not a drug. It's not a therapy. It's not a kind of curage. Vaccine is only antigen. It will induce your own immunity. That's why it's not correct to, it's even prohibited to give a vaccine for ill people. Because if ill person already have a live antigen, vaccine will do nothing. You will not make better. You will not help subject. Because vaccine will add antigen, maybe weakened, but still. Vaccine is always given to prevent disease when disease is not there. So we have another kind of definition. Vaccination. How to understand what is vaccination? Main number of people will say that it is injection of the vaccine. Not really medical definition. So vaccination is administration of a vaccine to raise the immunity against particular pathogen with the purpose to prevent the disease in the future. Not when subject is already ill. In the future, prevent. So when you will explain why do we need vaccines, please don't tell that we given for ill people to treat. No, it's only to prevent. And to prevent by raising the immunity, it means that your immune system should be ready for vaccination. So it is not a drug, but as any medical preparation, of course, it have some effects. The main effect is to raise the immunity. Vaccines should be immunogenic. It is the main indication to give a vaccine, to raise immunity against a particular disease. And your immune system will protect you, not a vaccine. your cells will induce immune response, produce memory, produce antibodies, and they can only help in the future. So remember that, please. And as any preparation given for a human, of course, it have indications, it have contraindications, it have side effects, it can have some adverse events, serious or mild. And of course, it's not the task of our department to explain every vaccine, to give you that list of indications and other. But you should know that almost every vaccine, not almost every vaccine, have one common contraindication. It is known hypersensitivity to any compound of the vaccine. Of course, if subjects have known history of allergy to some vaccine, that vaccine will be contraindicated. That's the only common. Main number of vaccines are prohibited for people with severe immunosuppression, with cancers, prohibited to pregnant women. But vaccinology is the main task of pediatricians. In pediatrics, we have that particular knowledge of every vaccine, when to give how, and etc. So I will not stop on that. It's the future department's topic. I need to give you common indications for vaccines, how to and when to apply vaccines. So as you can see, we have 5 of them. The very first one and the most important one is the scheduled vaccination. Scheduled vaccination means we have national schedule of vaccination. It is a scheme which is very unique for every country, and it indicates which vaccines should be used in that particular geographic area for every citizen of the country. So national schedule of vaccination is a cumulative data, because for long history it is already known which or what diseases are present in that area, in that country, which is very often here registered in high frequency. And of course the whole country should be vaccinated with corresponding vaccines to prevent that disease. So we have certain diseases which are pandemic, which are globally spread, such as tuberculosis, hepatitis B, some diphtheria, the same. And those vaccines are included in the national schedule of almost every country, because they are important in any part of the world. But we have some diseases which are limited to only a particular area. For example, in India, you have vaccination against Japanese encephalitis. In Russia, the disease is not registered, it's not included in our national schedule. Of course, in some Asia and other diseases are present, and we have some deviations. But some vaccines are very common, given in the same age in any part of the world. So, emergency vaccination. Till recently, it was not very easy to explain what is it, but now it's kind of obvious for everyone. So emergency vaccination is a type of vaccination when routinely some disease is absent. It's not known, or it's extremely rare, and none of the country vaccinate against that disease. But when the disease starts to spread as a local epidemic or a pandemic, surrounding territories should be vaccinated in an emergent manner to prevent spread of the disease. If surrounding countries will be immune, pathogen will have no options to spread to other territories. The perfect example now is that COVID vaccination, because disease was not known till recently, and of course no people around were immune. But when that epidemic started in China, of course surrounding countries was trying to prevent disease by vaccination, but it was too rapidly spreading disease, and of course we had no vaccines to prevent the disease, and it's happened. So what is post-exposure vaccination? Exposure means contact to antigen already occurred. It was not possible to predict the disease will happen, but exposure occurred, and you need to prevent disease in that one particular exposed person. So more often here we're talking about 2 diseases, tetanus and rabies. And rabies is not common disease. It's not so common disease neither in India nor in Russia to prevent scheduled vaccination of everyone, because rate is very, very low. and vaccination scheme is difficult. So vaccination against rabies will be done only if some person was bited. Dog bited, contact already occurred, and doctor need to vaccinate. The main task here is, the main kind of even goal here, both diseases, tetanus and rabies, they have pretty long incubation period. And that incubation period is enough to stimulate immunity. And to the moment when microorganism and pathogen will start to arrive in our body, our immunity will be already prepared for that. And tetanus in kids, scheduled vaccination is present, and you can be sure that if kid have good vaccination history, kid is immune. But in adults, even if some vaccine was done some years ago, but you cannot be sure that immunity is present right now. Because of very severe and hard complications, of course, it's easier and more reasonable to give extra dose of vaccine after injury, after some trauma. So vaccination of group of risk. Again, very limited indication. We're talking about some professions who are affected by one particular known disease. Dog hunters or veterinary doctors, again, rabies. They should be vaccinated in scheduled manner. Only that risk group. All laboratory doctors, those who are working with some known particular pathogen. Of course, it's more reasonable to prevent disease by vaccination before doctor will be exposed to that particular pathogen or antigen. And the last indication is pretty simple, vaccination of tourists. Again, if you're living in one country and you are going to visit another country, distant country, it's a very high chance that some disease in that destination country is present but not registered in your own country. And of course, local citizens in that country, they can be vaccinated by national vaccination, but you not. The same example, Japanese encephalitis, I'm not vaccinated against that. And if I'm going to visit India, it's much better for me to vaccinate before tour. The same problem if it is a doctor who is going to help some mission, volunteer mission in another country. Doctors are vaccinated against endemic diseases in that destination area before going here. So one more thing you should know, that first 4 indications, they're mainly covered by medical insurance of the country. Government is paying for that. You don't need to pay. The last one, if you go into tour to some tourism, of course, you should pay for that vaccination yourself, and you should check yourself which vaccines are required here in that country. So why I gave such a hidden name, vaccine nation? So mainly because it combines both definitions, vaccine and vaccination, but not that is the reason. We have such an idea, such a concept of collective immunity. To explain it easier, initially, naturally, we have a community I showed here, not vaccinated, neither ill with the disease, they all are susceptible. Immunity and those people are absent. And we have such a term as a threshold proportion. Threshold proportion, it is a percentage of population that must be capable of getting a disease in order for its spread. Means when everyone is susceptible, one person is infected. That one, for example, sick, became ill, but have no immunity. That person can infect anyone around because no one have an immunity. And the chance that people around will become ill is very high because they all are susceptible. And how many people should be ill right now to spread as a pandemic? That is a threshold proportion. Because initially we'll have one person, one very low rate. But when that proportion threshold will be reached, severe disease, severe pandemic can appear. To prevent that fast spread, to prevent pandemic, we need to have few people ill, and they of course will infect a lot of people around. So to prevent pandemic, we need to have that herd immunity. I don't like the term herd, but collective is easier. But we need to reach some amount of people who should be immune. They could be vaccinated, they could be ill before, but they should have immunity against a particular disease. And herd immunity occurs when a large proportion of community, we call it the herd, becomes immune to a disease. And if that one sick person is sick, whom can he infect? That one is immune, that one is immune, that one is immune, no one around. And the main idea of herd immunity, when that rate is reached, spread of disease from person to person became unlikely. Means we should have some minimal amount of immune people to stop the spread of the disease. And it's not even obligate to have everyone immune. Even if you have some, that person, here a few people, they have no immunity, they're susceptible. But no one around can transmit the disease. And those people are protected, not because their own immunity, because of nation immunity, because of that collective or herd immunity. And immune people around will protect that and that and that people. So we have kind of two contra concepts. How many people should be ill right now to spread the disease? It is threshold proportion. And how many people should be immune against the disease right now, not to allow pandemic. And at the very beginning of COVID-19, that herd immunity was claimed as a 75%. Calculations show that if 75% of overall population worldwide will be vaccinated, even we will have 25% susceptible. Disease will not occur as a pandemic. So when proportion of immune people will become greater than threshold, disease will decline. We will stop pandemic. That's why it is important to have nation vaccinated. Not every person even, but we should have a minimal rate of people who should be immune against the disease. And when that overweight of immune people against threshold will occur, it is known as threshold, herd community threshold. Of course, for different diseases, number is different. For some diseases, herd immunity is greater, some no. But main number of diseases are described good, and we know how many should be vaccinated or immune, how many we should not allow to be ill to win the disease. And in the case of some welfare diseases, especially if we will talk about some diphtheria, for example, or natural pox is a perfect example, we win that fight with the natural pox because people were vaccinated. We reached the rate of herd immunity. Microbe had no options, no ways to spread. And of course, it is possible to eradicate anthropanosis diseases which have only one host, human. If some disease present only in human, no other animals can be infected, no surrounding objects. It is possible to win the disease only by one simple measure, by vaccination. So there is a list of vaccines which are available now in the countries, in multiple different countries in the first column. And pipeline vaccines are vaccines which are in progress and research and development state. And that list was publicized in very beginning of 2020, and it was only some single reports about COVID-19 in China. Of course, those vaccines are not present. COVID vaccines are absent in that list. They were not known yet. But if you can see, we have that list of vaccines available. We have a huge number of diseases which can be prevented by only one simple measure, by vaccination. And almost the same list in pipelines. So that amount of diseases can be doubled up. And of course, it is very important to study and to know what vaccines are present, because some diseases are extremely dangerous. They cannot be treated. They have very high mortality rate, but they can be easily prevented by vaccine. As you can see in that pipeline vaccines, we have HIV vaccine even. Again, we will talk in more detailed manner in HIV topic and HIV class. But it is some experimental data on animals, and that vaccine was very promising. It was very effective in prevention of immunodeficiency caused by virus in mice. And if you can see that Shigella vaccine should be, in the end, Shigella vaccine we have here, it is designed by India. And India is the first country who going to register vaccine to prevent shigellosis in kids. The rest of the world have no vaccine. We had some to prevent dysentery only in Russia, but not very effective again. So maybe very soon we will have some changes in national schedule of vaccinations. So types of vaccines which are available. We have several types. The very first one and the most popular is live attenuated vaccine. Live vaccines are mainly done with the help of wild type pathogens which are weakened, or we say attenuated. So after administration of a live vaccine, of course, weakened virus or weakened bacteria can replicate in our tissues, but that microbe should not cause a disease. And live attenuated vaccines, they are repeating the natural way of the disease. And of course, they are the most immunogenic. They will provide you with the greatest and with the best immune response. And if a live vaccine is given in the form of injection, then generally they are effective following one single dose. But if we'll talk about some oral or other vaccines, polio vaccine, for example, given in a drop on the tongue, they're not so efficient, and usually we need for about 3 doses of the vaccine. But still they will have the best immune response. The only thing you should remember that, of course, if vaccine is alive, it's for sure contraindicated for subjects with impaired immunity, because their immune system is too weak. It cannot control even that attenuated strain, and they can cause, we say, vaccine-associated diseases. When after a vaccine, people will have disease. So we have the same polio vaccine as an example of a live vaccine. We have MMRV vaccine and others. So inactivated vaccines contain kill pathogens. The main benefit of that type of vaccine-associated diseases are impossible. But as far as they are killed, usually they are not so strong. They are not so good in immune response, and usually we require some booster shots and maybe even revaccinations. And one of the examples of killed vaccine is DTP vaccine. It contains pertussis, killed cells of Bordetella pertussis to prevent whooping cough. So next type of vaccine is subunit vaccine. Subunit vaccine is type of vaccine which includes the most immunogenic antigen of a pathogen, not complete cell or some viral part, only one immunogenic compound of a pathogen. And previously we usually obtained that type of vaccine by treatment of pathogens with different chemicals, but they're more allergic, they're poorly tolerated. And nowadays, main number of subunit vaccines obtained by genetic engineering. And subunit vaccines can be of several types. We have protein-based vaccines, which contain only protein antigen of a bacteria. And as an example, we have DTaP, acellular pertussis vaccine, because a lot of kids, not a lot of, but some portion of kids can have allergy on a cell component of Bordetella. So we have that subunit vaccine which contain only immunogenic part, not the whole cell. Polysaccharide vaccines contain polysaccharide molecules derived from external or outside layer of encapsulated bacteria. And now the main example is pneumococcal vaccine against Streptococcus pneumonia. We have several types, but polysaccharide vaccines are included as well. So conjugate vaccine is the modified polysaccharide vaccine, but when that polysaccharide compound molecule is fixed to a carrier protein to increase immune response, to increase immunogenicity. Mainly, that type of vaccine is used for infants, not to give a high dose of antigen, not to harm the baby. We given conjugate vaccines protein which is harmless. It will increase the immune response, but it is not so dangerous for a kid. And Haemophilus influenza vaccine is the type of that vaccine used nowadays to prevent kids' disease. And recombinant vaccine is the type of vaccine when only gene, some segment of gene, which is coding for immunogenic protein, is incorporated into the genome of another cell, which is absolutely harmless, some yeast cell, for example. And during replication of that yeast cell, that cell will produce antigen of dangerous virus. Here we're talking mainly about hepatitis B vaccine or human papilloma virus, because it's very difficult to inactivate. Virus is kind of, by nature, it's not active, it's not alive. But we need to be sure that virus will not harm human. And to prevent disease vaccine-associated hepatitis, only gene of hepatitis virus is inserted into the yeast cell. And yeast, of course, it is harmless, but it will produce hepatitis B antigens to induce immune response against virus. So toxoid, it's not really, again, I don't like to name it toxoid vaccine because it is toxoid, but anyway, toxoid vaccines contain toxin, exotoxin of a pathogen treated with formalin or formaldehyde and prolonged temperature exposure to keep immunogenicity but reduce toxigenicity. And toxoids are not toxigenic, they cannot cause toxic effect, but they will induce the same immune response as natural toxin. And of course we have DTE, diphtheria toxoid, tetanus toxoid as the part of DTP, for example. And combined vaccines is one of the most kind of easiest type of vaccine. It is type of vaccine which is obtained by different way, contain multiple antigens against different bacteria. The same again DTP vaccine, diphtheria toxoid and pertussis, tetanus toxoid and pertussis killed cell. One vaccine contains 3 antigens against 3 diseases obtained by different ways. So I will show it DNA vaccine. It is one of the most modern type. And I often say that smaller not mean worse. More primitive in structure or in organization, or just more not means less clever. And what do I mean? It happened that our cells of our immune system, very often appears much clever than we thought. And actually they are clever. We're thinking, cell, it's single cell, what can it do? It's just a cell. But no. During multiple experiments, it was found out that human immune cells can recognize not only structure of antigen, they can even acquire DNA of bacteria, perform transformation, and they will understand what antigen, the structure of antigen, which is coded by DNA. And it's appeared that it is enough to give DNA to our macrophage, for example, and DNA will produce immune response against antigen. Our antigen presenting cells will show structure of antigen, even they had contact only with DNA of bacteria. And of course, the type of vaccine is just easier to obtain, because to synthesize DNA, you need PCR. Just take PCR, multiply, make billions of DNA copies. That is the vaccine. Of course, it's not obligated to grow pathogen itself. That type of vaccine should be more safe, because of course, DNA of pathogen cannot cause a disease. And in that COVID, again, pandemic, we had few types of vaccines which are DNA based, and they showed their efficacy. They are used in humans already. So the next part of our lecture, again, vaccines prevention. Now you have serotherapy, seroprophylaxis. As you can guess, we will talk about preparations which are used to treat the disease or sometimes to prevent. What's the difference between immune serum and immunoglobulins? Immune serum is a serum containing naturally or artificially produced antibodies to a given antigen, and they can be obtained from animal or human source. Again, here the main idea, it is preparation which already have antigens. They are ready to fight right now. You don't need to wait until your immunity will work. That is a therapy, because antibodies can be applied right now to inactivate particular pathogen or some antigen. And immunoglobulins, it is very similar preparation, but it is a highly purified product made of pooled plasma of donors. And more often, to get one test tube of immunoglobulin, you need to collect blood from about 1,000 of donors. And as you can guess, of course, immunoglobulins are less available in routine practice. It's easier to obtain immune serum. but immunoglobulins are much more higher in activity. So in that topic and that section, I see two problems. The first one, always, every year, any group, always you are failing to answer that question. It's again, just think it's very simple, but how to obtain? You will have that preparation in exam and teacher will ask you how to obtain immunoglobulin against some gas gangrene, for example. Where can you get immune serum? And some students, those who I think don't like to think, they will say, okay, immune serum is always taken from donors, call a donor, take a serum in a volunteer, and you will get immune serum. Okay, I am a donor, I am a volunteer, take my blood. Do you remember the definition of antibody? What is antibody? Protein produced by immune system in the response to a contact with a particular antigen. To have serum against a disease, I should have a recent contact with the antigen. So if I'm a volunteer, I'm a donor, take my blood, will you find antibodies against that particular disease you need in my blood? For example, some gas gangrene. You have a person with gas gangrene, highly toxigenic disease, a lot of toxins. If you will take my blood, will you find antibodies here? Of course, no, because a subject should have previous response, previous contact with antigen, that particular antigen. And of course, if you remember the diagram I showed you, first exposure, second exposure, when IgM, IgG appeared and which amount, it should be recent contact, it should be a fresh contact, because antibodies will appear in high titer only in first or second week of the disease, then they will decline. So you need not just a donor, you need a donor at the second week after the exposure to antigen. So what to do? And your next option you given us, you students, it's killing me on the spot, and I sitting and having that face palm appearance, because you're saying, okay, let's find an ill person who is ill for about a week, and that person will have a lot of antibodies. Use him as a donor. Really? You really want to do that again? Guys, just think. You have a person who is ill. That person is in the progress of immune response. Those antibodies are required by that person needs antibodies to recover. If you will remove antibodies, that person will die. Or even I'm not talking about the fact that that person will have toxins in the blood, not only antibodies. Person is naturally ill, and blood contains a lot of active toxins, and you will give that mixture to your ill person. So you have another brilliant idea. Brilliant. You're saying, okay, human is not very good, yes, but let's take animal. You know, like animal, don't mind losing it. Guys, don't do that, like, please. Animal, the same again, where are you going to find that animal? Or you should keep a bunch of animals, and some animals should be ill in the second stage always, in the second week. It's not working like that, you know, like we have another thing. That one, hyperimmunization. Of course, at the very beginning, people try to take blood of ill people to perform some experiments. And nowadays, any preparation should be absolutely safe. We're not using ill people, ill animal. No, we have hyperimmunization. The term is extremely important because hyperimmunization procedure is used to obtain immune serum and immunoglobulins. I will even mark like that, hyperimmunization. Any teacher, any class or exam ask how to obtain immune serum or how to obtain immunoglobulin. Just say, we can obtain them by hyperimmunization. Hyperimmunization, it is presence of larger the normal number of antibodies to a specific antigen. Not just any, if you take any donor, to a specific antigen, you should know which antigen is here. And to develop hyperimmunization state, of course, you need a donor. It should be a volunteer, not ill person. It should be absolutely healthy person to give you good, strong, potent immune response. And the donor should be stimulated with excessive amount of one or few known required antigens. Not the whole pathogen should be there. A live bacteria producing a set of unknown factors. You should take one known antigen or few known antigens. And of course, they should be safe. You should not overload your donor. You should not kill that donor. Not the toxin you should use. We have vaccines. You should vaccinate donor with safe preparation, with excess amount of the vaccine. Again, not that scheduled vaccination is used. It's not any person around. That donor should be vaccinated with the purpose with one known vaccine, but an excess dose. And immune system will produce excess amount of antibodies. Person don't need them because person is healthy. And the donor volunteer will donate antibodies and you can use them. Of course, you will purify them. And in medical facility, you will always have that commercial preparation. And it will be labeled what is it and how was it obtained. If it is a vaccine, it will be some recombinant vaccine. If it is a serum, you will have some human serum obtained by and that. I really hope it will be easier for you now to understand that immune serum and immunoglobulins are preparations obtained by hyperimmunization of donors with one only purpose, to obtain excess amount of antibodies against particular disease. Don't search for ill people, don't search for any person around. It is a specific procedure. So, as I said, preparations can be of two types. They can be of two origins, animal origin and human origin. But in medical practice, we're using slightly different word. We're using homologous and heterologous. And again, it is a second problem. I said I see two types of problems. First one, you're always confused about the question how to obtain serum. Second, we're asking, is it homologous or heterologous? And you are confused. Or we're asking, what is homologous preparation? What is heterologous? And you're saying, thinking, you see that logos, immune, serum is the same. Logos, immune. So the difference is in the first only 4 letters, or 5, some 6. Homo and hetero. That's the difference. And you know that immune serum was used because of antibodies, and you're saying that homologous serum contain the same antibodies and heterologous serum contain different antibodies. I cannot say it's completely incorrect, but what do you mean? In one tube we have the same antibodies, in another we have different antibodies. It's not really like that. So again, as I mentioned, in medical practice we have two chains of one problem. You have ill person who needs antibodies right now, and you should have some donor, volunteer, who will donate antibodies. We have recipient and we have a donor. And we said the donor can be of two types. It can be animal, it can be human. And actually, the same with the recipient. Of course, in human medicine, you are human doctor. You will deal with humans, but we have veterinary as well. And they have the same problems. They use the same serum for treatment. So maybe you guessed already now. Homologous immunobiological preparation is preparation obtained from the same animal source is going to be injected to. If your donor and recipient are the same, preparation is homologous. If donor and recipient are different, it is heterologous. And what is the main problem here? Again, immunoglobulin even, it's not really important. In serum, more important it is in serum. Because what is serum? You all know that it is not only antibody. Its preparation contains set of different compounds. And of course, you will inject all into your subject. And if you study in detailed manner, in particular manner, composition of blood in horse, in human and horses are mainly used as a donors of immune serum. Kind of a set of list of compounds, participants of the blood. It's pretty identical. You will have the same types of blood cells, you will have the same vitamins, the same classes of antibodies, the same even common structure of antibodies. But still, animals have slightly different structure, organization, and of immune system. And antibodies, kind of by concept, they're the same, but they can have slightly different structure. Slightly, but different. And you already know that our immunity is able to identify that even slight difference. And of course, if you will give animal serum to a person, some compounds of that serum can be recognized by our immunity as a foreign, and the subject will induce severe allergic response. We will talk a little later in current lecture. So when in classes we given you a test tube and asking, is it homologous or heterologous serum? You trying to find the legend. It should be, you think that it should be shown, is it homo or heterologous? No. In the test tube, always you will have only donor. Who was the donor? Is it horse serum or human serum? But manufacturer do not know how you're going to use that serum. Maybe you will use to treat human, maybe veterinary doctor will use it. So the main idea, if you asked about how to treat human and you see that serum is obtained from human, of course it is homologous. If you see that it is horse serum and you go in to treat human, it is heterologous. But if you will take horse serum to treat horse, even horse serum for horse will be homologous. So again, just think, in human medicine, human serum is homologous. So okay, few words again here. Serum and immunoglobulins are used for treatment. I said we have three types of compounds, three types of preparations: vaccine, serum, and diagnosticums. Vaccine, we explained, is used for prophylaxis of the disease, but as well it can be used for hyperimmunization to obtain immune serum. So vaccines have two main goals. The most popular, the most frequent one is prevention of the disease in community in some people, but as well it can be used to apply immune serum or immunoglobulins. And immune serum immunoglobulins are used for treatment or prevention of the disease, but mainly, again, we're talking about people who are already exposed and they need to have some protection right now. During private microbiology, we will give a more detailed explanation. But vaccine contain antigen to induce immune response, and your immunity should do it before exposure. Immune serum immunoglobulins, they contain antibodies used right now to give for a person who is ill or exposed already. Vaccines, antigen, they will induce active immunity, artificial. Antibodies are present in serum and globulins. Antibodies will provide passive immunity, and again acquired. Vaccines, active acquired immunity; immune serum and immunoglobulins, passive acquired immunity. In both cases, it will be, it can be artificial in the case of vaccine. It can be natural or artificial in serotherapy. If we're talking about mother to fetus, sharing of immune globulins with the mother will be natural, but more often serotherapy, seroprophylaxis, it is artificial type of immunity. So, sorry, now next slide about that hybridoma technology. So, we're talking a lot about antibodies, and we will discuss a lot of things about antibodies. Last lecture, I told you that we have ELISA, which is based on the use of monoclonal antibodies. And a lot of antibodies are required, and nowadays we have that hybridome technology. We have some directions, we have some experiments already in human even medicine, how to use monoclonal antibodies in the treatment of a particular diseases. in the treatment of autoimmune diseases, in the treatment of infectious diseases. And it's open secret that the medicine is very fast progressing now. A lot of technologies are available, and very often we need a lot of different antibodies against a particular disease. So of course, previous years they were obtained by hyperimmunization mainly of animals, but huge number of animals are required. It's first expensive to keep so huge number of animals. Second, it's difficult to immunize every animal to keep to wait after immunization. You need to obtain the serum, purify, a lot of problems we have. And of course, I'm not talking about ethics. A lot of animals are killed just to obtain some antibody to perform maybe unsuccessful experiment. A lot of people are not happy with that. So nowadays, problem is partially solved by hybridoma. What is it? Again, in books, and especially some not student book and medical and scientific books, it's very difficult to understand. That style of explanation is not for students. So I will try to explain in few words. Hybridoma, even the name vocalize, hybrid chimera. So we need to make a hybrid of two cells. You need antibodies. As you know, you need to be plasma cell, and it should be stimulated by immunity. So as you know, spleen is the main organ accumulating antigens, and it is the main organ responsible for cell activation, for activation of immune response. So you need to expose the mice with an antigen. When immune response is activated, you will take spleen cells, spleen of the mouse, antibody producing B lymphocytes we need. They are isolated by different, some centrifugation or other different measures. B plasma cells are isolated, and you need immortal myeloma cell. It's kind of a cancer cell which can multiply immortally. It have unlimited time of life. And they need to fuse B plasma cell and that myeloma cell. And you will have a hybrid with two options obtained from both parents. It will be immortal, and it will produce your required antibodies. And it is one single cell. Of course, every antibody will be absolutely identical to previous one. And you will have a pool of antibodies, a clone, single clone, one clone of antibodies. That's why monoclonal. And of course, that pool is unlimited. Your only task is to keep the cell alive. It is immortal, so you need only to kind of feed the cell. It will secrete a lot of antibodies. And you can use that antibody for any purpose you need. You need to label them with enzyme and apply for ELISA. You can label with some fluorochrome and use it as immunofluorescence diagnosticum. And again, nowadays we have experiments how to apply antibodies in the treatment of the diseases. But till nowadays, again, we have one very significant problem. There is a problem, mouse. Because mouse, human, we are two different instruction organization. And mouse antibodies are not always compatible with the human. And human myeloma cell is not very stable, so we have some problems how to get human hybrid cell. But some experiments are going on, and nowadays, and you already will meet antibodies, monoclonal antibodies used for treatment of multiple diseases. And that technology is really promising, and it's in progress. So the last type of preparations we need to say, diagnosticum. As I said, we have two types. So in common idea, diagnosticum is a standard preparation contain known antigen. Standard preparation contain antigen. It is diagnosticum. But the main difference with the vaccine as well as a standard preparation contain antigen. But vaccine is used to raise immunity with the purpose to prevent disease, and diagnosticum is a preparation used for diagnosis of the disease. We have differences in method how to obtain. Vaccines can contain, and they should contain, only immunogenic compound. In diagnosticum, more often we have killed microbial cell. It is always complete the whole cell because it is used for lab. In lab, diagnosticums are mainly obtained by treatment of a live microbe with formalin. It is toxic for human. You cannot inject it into the human. So it is used for action of agglutination mainly. And here it is Russian Russian photos I gave you. But you can see that heme agglutination here and that. You have heme agglutination. And first here, for agglutination test, some diagnosticum for action of agglutination. If you see that it is agglutination, please, guys, don't give it to subject. Don't inject it into human. Any type of diagnosticum, if it is mentioned, diagnosticum, it is for laboratory, for in vitro test. Any vaccine will be labeled vaccine. DTP vaccine, BCG vaccine, MMRV vaccine. If you see the name vaccine, that is really vaccine. It is used to prevent the disease. If you see the name diagnosticum, diagnosticum is for lab test, main direction of agglutination. If you see serum immunoglobulin, it is preparation containing antibodies used for treatment of the disease. And we have a set of, very small set of diagnosticums, but they are for in vivo diagnostics. And they are ends on IN. Next, in private microbiology, we will discuss. You will have tularin. It is mixture of killed Francisella tularensis cell, used for allergic skin test. You will have tuberculin, Mantoux test, or some Perquet test, another. That is for human use. If you have IN at the end, it is to apply for injection, in injection, tularin in injection. If it is diagnosticum, it is for lab agglutination test. Please remember that is easy, but remember and think. And when you will be provided with some test, you put name of the test. First think, read, then answer.